will speak about evaluation of rabbit as a laboratory model for bovine viral diarrhea virus. Bovine viral diarrhea virus is a very important pathogen of cattle, which can naturally infect a wide range of even-toothed ungulates. Bovine viral diarrhea is a member of virus within family Flaviviridae. Actually, viruses have been classified into two genotypes, genotype 1 and genotype 2, according to genomic characteristics. And each one has been divided into two biotypes, cytopathogenic type and non-cytopathogenic type, according to their behavior on cell culture. We have also to mention that high viral strain can spread, excuse me, with his pointer. He can spread to almost every organ. Highest number of antigen found 6 days post-infection. And the primary replication actually occur in lymph nodes and tonsils. We have to say that it's a lymphotropic virus. Clinical symptoms including fever, leukopenia, thrombocytopenia, depression, anorexia, oculonasal discharge and oral erosions and lesions, and occasionally diarrhea and death. Animal susceptibility, mainly cattle, but as I said at the beginning, it can infect all artidictulous or even toothed ungulates like pigs, sheep, goats, alpacas, deer, reindeer, and bison. Our problem is we don't have standard lab host to facilitate our work in the virus. And actually our point of interest in this virus, not from that point that he is bovine viral diarrhea of cattle, actually we use it as a surrogate model for hepatitis C virus. And we will speak about this in my next presentation. We have to just focus on this short review. At the beginning, when they discovered this bovine viral diarrhea virus, the Becker et al. Tried to make some research to discover range hosts. And they inoculate many animals, non-artidiculous like horses, cats, dogs, several small laboratory animals like guinea pigs and mouse and rabbit, and breeded chicken egg. And finally, he concluded that the only non-artidiculous animal who can propagate the virus is rabbit. Also, another scientist worked in this area, and he concluded that spleen homogenate from rabbits can infect cattle, and also bovine viral diarrhea virus can be serially passed between rabbits and cows. And it was 1996. Since this moment, no one isolate bovine viral diarrhea rabbit or succeeded in infection of rabbits with bovine viral diarrhea virus. Then another teamwork with Spackfun et al. From Switzerland start to work on this. And they got this hot field Scotch strain MRI 103 for experimental infection of rabbit. And they concluded that are susceptible to infection, that rabbits are susceptible to infection by bovine viral diarrhea virus, but infection doesn't cause clinically apparent disease. And they care about this problem from epidemiological reason, They were trying to understand if rabbits can act as natural reservoir of the virus or not. And I delayed this a bit, this Froelich and Streich's German team. And in 1998, they discovered that 40% of free-ranging rabbits in Germany are seropositive to Nadel strain bovine viral diarrhea virus without any virus isolation. Actually, this Nadel strain is laboratory strain, laboratory adapted strain, Natural American Disease Laboratory Institute strain. Our aim of the work, complete evaluation of rabbit as susceptible lab host for bovine viral diarrhea virus. We have the virus fully identified with sequencing of 5-UTR lesion, 3 passage on MBDK cells, titration, filtration, and its concentration was 10 power 6.6 tissue culture effective dose 50 per milli. MBDK cells supplemented with bovine serum, of course inactivated. Animals, 33 New Zealand white male animals, 60-day-olds, engaged, fed, and received water ad libitum, received daily elimination, 16 hours of light, and supplemented with coccidiostatic orally in water and ivermectin subcutaneous and anti-mange and we kept them for 1 week just under observation. After this, we start our first experiment, virus adaptation. 3 rabbits created with 1 milli of 10 power 6.6 tissue culture effective dose 50 of virus. 6 days later, animals have been immunized and the spleen collected and homogenized and filtrated. From this filtrate, we made successive 3 blind passages of the virus inside rabbits using splenic homogenate and puffy coats, followed by virus isolation and retitration from splenic tissue. Animals start to show signs of depression and off food and diarrhea at second passage, not first passage. First passage was asymptomatic. Animals was apparently normal. But starting from second passage, they start to have some diarrhea, self-limiting diarrhea, and off food. On third passage, one animal died with nervous manifestation. Then we also re-isolate the virus again and prepare it for next experiment. Second experiment is an evaluation of rabbit as a model for bovine viral diarrhea virus. We have 2 groups, Group A and Group B. Group A is control negative group. We inoculated with just only media without any virus. Group B, bovine viral diarrhea infected virus, inoculated with 10 power 6.6 tissue culture effective dose 50. Then on 3rd and 5th and 7th and 10th day, post-inoculation, 6 animals from each group was euthanized and sampling occurred for virus isolation. RTE, PCR, histopathological study, and clinical pathological examinations according to Conferenceseries et al. Results, clinical signs, mild depression during time of experiment, dropped ear, inhabitants for the first 3 days, then followed by diarrhea starting the 5th day till the end of experiment. Nervous manifestation and convulsions followed by death in one of animals. This video for one of animals, we can see that he has nervous manifestation like convulsions and oronasal discharges. This is my hand. And also, I didn't mention that we have, these animals are vaccinated against hemorrhagic septicemia and pasteurellosis. This is diarrhea. Post-mortem examination, we found that cecum become very enlarged very much with congestion of blood vessels, mesenteric blood vessels. Same congestion of mesenteric blood vessels and of intestine, small intestine. Lung, this is normal one. This is infected one. Normal. Brain, hemorrhage and congestion. Spleen, shrinkage, mottled, pale, friable, and it's a very pathognomonic for this virus. This is lymph node, edematous, actually very big, and hemorrhagic. Then virus isolation. On 3rd day and 5th day and 7th day, actually in 3rd and 5th day, we re-isolate the virus. On 7th day, virus disappeared from 2 animals, and also we isolate it only from 1 animal on 7th day. After 10 days, complete clearance of virus. Quantitative real-time PCR for titration of the virus, to follow it up every 3 days, 3 on 3rd and 5th and 7th, to follow titer of the virus. This graph to describe increase in titer of virus from 3rd day start to be clear with 69 tissue culture effective 50 per gram, reach to maximum on 7th day with 190 tissue culture effective 50 per gram, and start to decline on 7th day. Then disappear after this. About clinical pathological study, hematology, we have decreased RBC's number due to hemorrhage, same as hemoglobin. This is control 1. Severe thrombocytopenia, severe leukopenia, lymphocytopenia. Sorry. have here two photos for liver, for histopathology of liver, but it seems that disappeared, some problem happened. But this photos shows that congestion and dilatation of hepatic sinusoid with dissociation of hepatic cords, pericentral inflammatory cell infiltration, is vascular degeneration in some hepatocyte, and we confirm these results with immunohistochemistry using streptavidin biotin immunoperoxidase. As we see that we have here our antigen with a specific monoclonal antibody against bovine viral diarrhea virus. This is lung of rabbits with peribronchial mononuclear cell infiltration. Also, we result confirmed with immunoperoxidase, as we see. Spleen show marked lymphocytolysis with appearance of underlying reticular mesh. All these free spaces. Also result confirmed with immunohistochemistry for demonstration of antigen. Sorry. This is lymph node, section in lymph node, also showing lymphocytolysis and appearance of reticular mesh, and confirmed with immunohistochemistry. From these results, indicates that rabbits are susceptible to infection with bovine viral diarrhea virus, NADL strain, and that infection causes transient infection, and the clinical pathological picture of the virus infection in rabbits close to that of natural host. Let's move to conclusion. Experimental infection of rabbit with bovine viral diarrhea can be achieved with rabbit adaptive NADL strain 3 passages. Clinical picture of experimental VVD infection appear as depression, impotence, and self-limiting diarrhea. Some animals show nervous manifestation and complete clearance of virus intensity. Thank you.
Thank you very much for this nice talk. I think it's this short time of incubation in rabbits, we have no problem with secondary infections of bacteria.
Actually, my interest in this point, not problem of bovine viral diarrhea infection is his ability to make resistant infected animal. And this area is not my interest, actually. My area, I want to make that lab model for transient infection of bovine viral diarrhea virus for further researches on this point as antiviral. This is what my interest, I'm not caring with resistant infection. Anyone have any question?
Yeah, with the strain that is now adapted to be able to grow in rabbit, is there any difference in the genome sequence?
Actually, no.
No, you've done the sequencing.
Yes, 5-UTR genome sequence have been done in America by head of department of virology, actually who provides this virus to me, and no mutation at all. Actually, this virus just have something like missing because it was laboratory adapted strain, so no one think to use it in animals and experimental infection.
Okay.
Just once, maybe in 1970.
Yeah. But you might have to do the whole sequencing of the whole genome, not just 5' UTR, because in the virus that we work with, when it adapts in animals, in mice, there are mutations in the genome, certain amino acids.
But I also mentioned that 1998, German team here, in free-ranging and wild rabbits, they found 40% of rabbits with antibody against specifically this strain.
Okay.
But actually, so but it's a good idea. It can be.
If you find, you can publish a paper.
Yes, yes. Yeah.
Hi, thank you. How do you think this correlates with the real process of infection? Because you have to adapt the strain, and in nature it will not happen. So, because the idea, if I understood it well, is to use it as a model. How do you make this contrast between the lab model with adaptation with 3 passages and the real thing in nature?
Actually, I have to believe in this because actually I can't make my experiment on cows. We don't have this money, we don't have this space, and we cannot take this risk because it's a notifiable disease. It's not a symbol. So we have to make our own lab host for this experiment. And my point of interest, I will explain it next presentation, about antiviral activity. And we want to test it in vivo, especially on bovine viral diarrhea virus, because it has antigenic similarity with hepatitis C virus. And this is our only option to do this. And actually, we spent 2 years in this adaptation. I said it in a few minutes, but actually, it takes 2 years.